Welcome back to the demonstration on the analysis of DNA sequencing data. In this segment, we will be performing variant calling. We will be continuing with the same example we were working on in the previous segment. If you have not used your Galaxy account since last lesson, all your history should be saved in your default history list. There are many programs for variant calling. GATK, VARSCAN, and Freebase to name a few. We will use the SAM Tools BCF Tools package, an easy to use and popular choice for variant calling. So let's head back to Galaxy. In the last segment, we had aligned our reads to the HG19 version of the human genome, producing a BAM file. The SAM or BAM alignment formats are read-centric, in that every line in the file refers to a read. BCF Tools, in the first stage, converts the data to a format known as a pileup, where every line represents a position in the genome instead. A pileup is essentially a column-wise representation of the aligned reads to the reference at the base level. The pileup file summarizes all data from the reads at each genomic region that is covered by at least one read. We will now use the BCF Tools MPileup program, which generates a file containing the genotype likelihood at each genomic location covered in the BAM file. Begin by clicking on the VCF BCF link on the left and scroll down to the BCF Tools. Here's BCF Tools, and we're looking for the MPileup. When you find it, click on the BCF Tools MPileup, and it generates either a VCF or BCF containing genotype likelihoods for one or multiple alignments. Click on that. Okay, now it opens up the choices, the parameters for the BCF Tools MPileup. Now here we're working on a single BAM file, and our input should be there by default. If that is the last BAM file that you have running, that's our BAM file that we had from last session. And here you will choose the reference genome to be HG19 that we had aligned to in the previous session. And our output type, we could output a compressed BCF, but that would be a binary format. So in order for us to be able to view the file, we're going to choose the uncompressed VCF file, and then click on execute. And then you'll see it starting to run the program on the right-hand side. Most variant callers, including the BCF Tools package, produce a standard file format known as variant call format, or known as VCF for short, also known as BCF in its binary compressed version. Now let's see, the MPileup program has completed. Let's take a look at the VCF output file by clicking on the I next to the results section on the right-hand side. Now the VCF format consists of a header section and a data section. The header section each line begins with a pound sign. The header section contains some information about the file and the parameters used to produce it. The header also specifies what information is stored in the fields Info, Filter, and Format columns, as this is different for different variant callers. Now, before we look any further at the VCF file, it is important to remember that we have only performed the first step in the BCF Tools pipeline. As mentioned earlier, the MPileup generates genotype likelihoods at each genomic position that is covered by reads. Then you need to use the BCF Tools call program, which makes the actual variant calls. So roughly, all we've done so far is turn the BAM file into a VCF file. We will now run the BCF Tools call SNP indel variant calling, part of the BCF Tools package. On the left side of the screen, you'll click on VCF BCF, scroll down until you reach BCF Tools. Here's BCF Tools, and look for BCF Tools call. Should be right here. Click on it. Okay, the input will be the output from the previous MPileUp program. And the only other change you would have to make is click on Input and Output Options. Okay, and here we would choose Variance Only, choose Yes, and also change it from compressed BCF to uncompressed BCF so we could actually view the file, and then click Execute. The BCF Tools call program has now completed. Let's examine the output from the call program. But first, notice that the number of output lines from the MPileUp number about 580,000 lines versus the number of output lines from the BCF Tools call program, which lists only 309 lines, as now we are only including the suspected variant sites and not the entire area of coverage. So we can now look at the VCF resulting file by clicking on the I next to the BCF Tools call results. Okay, the VCF file should now open up in the center. We will now concentrate on the data section below. This is the header section, as you see the 2 pound signs before each line. And let's go below, and now we will look at the data section. The data section has several fields. Let's start with several of the fields, chrome and position. These describe the variant's genomic location. The next field, ID, would contain the identifier of known SNPs in the case when the VCF file had been annotated. Ref describes the reference base or bases in the case of an insertion or a deletion at the given position on the reference sequence. Alt describes the list of alternative alleles at this position. The next field is the qual field. Qual is a quality score giving the confidence in the correctness of this variant call. And basically, the higher the score, the more likely this is actually a true variant. That's then followed by filter, which would contain a flag basically indicating which filters this variant passed. Followed by the info field. The info field is a key value pair field, which can contain multiple fields separated by semicolons, and it can contain all kinds of information depending on the variant caller used. One example is DP. DP is depth. So for instance, DP equal to 196 would mean that there are 196 reads supporting this variant call. You can see the documentation on VCF files for the other different flag value pairs. Genotype information is given in the next 2 fields. First, a format field is given specifying the data types and order in a string that is separated by a colon. This is followed by 1 field per sample with colon-separated data corresponding to the data types specified in the format. Now, genotype is encoded as allele values where 0 is for the reference allele, 1 is for the alternate allele, and 2 in the case where there's a second alternate allele. Thus, in the case of a diploid genome, a genotype of 0, 0 would indicate a genotype that is homozygous to the reference. A genotype of 0, 1 would be heterozygous, and a 1, 1 genotype would indicate a homozygous to the alternate. In addition, you see here the PL field. The PL field is actually a likelihood value of the genotype, and the PL field contains 3 numbers, and these 3 numbers correspond to the possible genotypes 0, 0, 0, 1, and 1, 1. And these values are so-called normalized so that the most likely genotype will be given a 0 in the Phred scale. So this is just a taste of a VCF file. And if you'd like further information, once again, here's the URL for the VCF specifications. So how many variants do we have in our VCF file? What are the different types of variants? The VCF toolbox contains tools for this kind of analysis. Let us look at the BCF Tools Counts program. Again, you can find it under VCF BCF, BCF Tools, and scroll down until you find the BCF Tools Counts program. Click on the BCF Tools Counts. It should open up, and the only thing to put in is our VCF file that we have from our BCF Tools call analysis, and that should be the default if that's the last thing you ran, and then you would click execute. This will now run on the file and give us the count of how many variants there are. Let's click on the eye to see the output from the BCF Counts program. As you can see, we have one sample in our BCF file, and this sample has 257 SNPs and 52 insertion deletions for a total of 309 sites in our BCF file, which is what we saw previously from the BCF Tools call. Now, to identify from these SNPs what are the true SNPs, non-artifact SNPs, is often not a trivial task, and therefore generally SNP calling is followed by some type of filtering step. Now, just to be clear, variant filtering is not trivial, and it is often difficult to tell variants and artifacts apart. There are several rules of thumb. First of all, if its proximity to an insertion deletion, because often in an area of insertion deletion, it is difficult to determine a correct alignment. Therefore, it might be an artifact. Also, the number of high-quality reads supporting the site, and in addition, the quality score given by the variant calling program. The variant callers provide a quality score, a qual column, which we mentioned earlier, which gives an estimate of how likely it is to observe this call purely by chance. So an easy way to filter low-quality calls is to just remove all variant calls that are below some threshold. So let's try that right now. We can click on, under VCF BCF, there's something called VCF filter. So let's click right now on the VCF filter. The input for the VCF filter would be the VCF file that we had generated using the BCF Tools call program. For the sake of this demonstration, we will look for those sites that have a qual greater than 200. So here, specify filtering value, and here we'll look for qual greater than 200. And then click execute. The VCF filter has completed. Let's click on the link. And what do we see? This now has 247 variants listed, reduced from, if you remember in the previous, from the previous file, from 309 sites. Let's click on the eye and see the VCF file. And let's try and see if the variant that we examined in the last exercise was detected. If you remember, it was on chromosome 10 at 96,541,616. So we'll just do a search on the file for 965,41616. Indeed, the site that we examined in the last exercise was detected by the variant calling pipeline. And if you remember, it was a G in the reference and an A in our sample. And what was the genotype of our subject? If you remember, the genotype was a heterozygous genotype. And indeed, here we see the 01 or the heterozygous genotype of our subject, as we saw in the previous segment. So to summarize, in these three segments, we've seen how to align the raw reads, DNA sequencing reads, to a reference genome. We learned how to generate variant calls from aligned reads, how to interpret the various file formats used in storing reads, alignment of reads, and variant calls, as well as visualize the data using the UCSC Genome Browser. In the coming lessons, you will learn how to prioritize and assess the functional significance of these variant calls and how they relate to human health.